stuff on population genetics kind of at the end of class, and I had a few questions about it by email. So let me just summarize briefly a few things here before we go into the next kind of extra topic section. So I just want to point out that one obvious type of problem are these migration problems, and the most obvious way to view this is in terms of these colored boxes. Imagine, a certain number of individuals from one population and a certain number of individuals from another population combined to make a new population, right? And in that new population, you have to calculate, let's say, the new allele frequency, which is going to be dependent on the proportion of individuals in the yellow box and in the blue box. And variants of this problem would be, well, what if there's 3 populations or 4 populations, or one migration event, then another, or something like that. But all those things really don't change the basic concept of this type of thing, which is to kind of add things together based on the proportion with which they contribute to the new population. Okay? And then the other key thing is just to be careful when you do and when you don't apply party wonder to these types of problems. So that's that. And then just briefly, again, something about these fitness problems. The basic idea in this slide, to summarize or to review, is that if you need to calculate the fitness value, one way to do it, if you were to be given number of offspring, is to compare the offspring of all the different genotypes, the number of offspring from all the different genotypes, to those genotypes which had the maximum number of offspring. So somebody ought to be set, at least somebody ought to be set at a level of 1. And in this problem, some artificial problem, this genotype had the most number of offspring, so that's set at a level of 1. And don't make the mistake in these types of problems of summing all of these and dividing everything over the sum of all of those. In that case, you'll never have a genotype set at a level of 1. So just one caution in those types of problems. And then almost the last thing that we went over was this table. And really, this is a plug and chug operation, and this is the extent of it. I mean, we're not going to get more complicated with all these other mathematical things in the textbook. I mean, we're trying to cover a lot of chapters. We have to cover classical genetics and molecular genetics, and that, so on and so forth. So we can only do so much. This table, I think, illustrates this basic idea that through selection or through fitness, you can change allele concentrations or allele frequencies through time, right? And just to remind you very briefly, the idea is you start out with the population, which in this case is in Hardy-Weinberg equilibrium. So we're given some P and Q values here, and we plug those into P squared, 2PQ, and Q squared. That sets the problem up. You have allele frequencies, you have genotype frequencies, assuming the starting population was in Hardy-Weinberg equilibrium. And you just have, I know it looks like a mess, but it's pretty straightforward. You have 3 columns. You have to deal with this column, and this column, and they're all genotypes, homozygous or heterozygous. And in this particular chart, we've been told a fitness value for each of the genotypes. In other problems, like I said, you may have to make some calculation about that, but in this one, they just give it to us, and that is the fitness of this type is 0, fitness of this type is 0.4, fitness of this type is 1. Somebody has to be 1 in these problems, and you multiply your way through. So the genotype frequency of A1 times 0 is 0, 0.48 times 0.4 is some value 0.19, 0.16 times some value, in this case 1, is still 0.16. And so we arrive at an initial apparent genotype frequency for all of the genotypes after we've applied the fitness constraint. And sometimes this is an issue, but the problem is the genotypes that we end up with after we apply the fitness constraint don't make the population into a new 100%. So you have to add up all these frequencies, and you have to compare 0.19 to what all is in the population, right, which is 0 plus 0.19 plus 0.16. And you have to compare 0.16 to whatever's in this new population. And that's why we have these values over here of 0 over 0.35, 0.19 over 0.35, 0.16 over 0.35. That normalizes it, the new population out of 100%. And so we get some new genotype frequencies. It may be, like I said, the population is smaller, so be it. Nonetheless, of this new population consisting of 100%, we have about 54% heterozygotes, 46% homozygous A2, A2. Okay, so that's kind of usually what you're trying to do in these problems, and the rest of it in this kind of box here just calculates allele frequencies using that one approach where we look at genotype frequencies of the homozygote plus half the genotype frequencies of the heterozygote, and so we have determined new allele frequencies. Okay, so this is kind of where we stand, or the level of difficulty I think is appropriate for these types of problems. It gives you an idea what's happening when we apply some selection criteria, and it gives you an idea that allele frequencies change through time. Now this problem is really dramatic, right? I mean, this is 0 and this is 0.4. If A1 is a dominant allele, there's just an absolutely, unbelievable selection against this allele, and a very strong selection against this allele, even when it's in a heterozygous form. And so even over one generation, you see this, huge effect on allele frequencies in the population you begin with and the population you end up with. In reality, we would probably say that for, most alleles or nucleotide changes, we would have selection criteria that would be really, really, really small, right? Maybe 0.9999, just some value like this. Something impossibly small, so that when you work it over one generation, you'd hardly see any effect. But if you applied a fitness constraint like that over 100 or 1,000 or 1,000,000 generations, you would start to see a change in allele frequency. Okay, so that's, I think, good for this. And then I had ended here and just went over briefly this idea that we could kind of catalog all sorts of arrangements where we say the fitness of certain genotypes is equal to 1 or less than 1 and so forth. And we've got a whole table or a whole list of possibilities here. In the first one, having allele 1,1 is equal to fitness of 1, heterozygote equals 1, and allele 2,2 equals 1. So everybody is equally fit in that particular scenario. And all we do is kind of run through various alternative scenarios. This one, for instance, is allele 1,1. Let's see which one's dominant. Let me pick another. Maybe I switched that. Okay. This one, less than 1. Okay, so this is like the problem that we just saw, right? We have allele 1,1, which we'll say is the dominant allele, heterozygote 1,2. Both of those are selected against, so they're less than 1. That's what we saw in the previous chart, 0 and 0.4 fitness values. But being recessive, homozygous 2,2 equals 1. So each of these describes a different scenario where you are either selecting for or selecting against various genotypes. The table goes through that dramatic example, but in reality, probably the selection or the fitness values would be, very, very close to 1. So and the note here is just to say that we suppose that these schemes impact all the alleles of all the possible 20,000, let's say, genes, let's say in humans or mouse or whatever, all of which come in all these allelic forms, but there may be just exceedingly small fractional fitnesses or fitnesses that we can't even, imagine or determine. And the other question here is maybe these kinds of ideas also apply to all the differences between different genomes. We don't know, right? It's impossible to measure those things, but there's lots of sequence changes from one mouse to another mouse to another mouse and so forth. So it's an interesting idea. And overall, the overriding point is frequency of alleles in the population and the types of individuals that can be made are going to change through time. So I made this point before, but at population, in some population at time 0, you have certain allele frequencies for all the possible genes that make up that organism. And the types of individuals that you can make from that population of alleles is basically determined by kind of picking things out of the vat, right? So maybe you pick 20,000 times, get 20,000 genes, and you can make something. And what you get is going to be determined by the frequency, the chance of picking certain alleles at time 0. But if you go 1,000 or 10,000 or 100,000 generations later, allele frequencies change through these types of things we've been looking at, right? Migration, selection, mutation, et cetera, et cetera. And so allele frequencies will be different many generations later. And so the types of organisms that you're likely to pick out or to create, their characteristics, are going to be different at a later point in time. That's basically the idea here. Okay, so good. A few notes about this chapter. I made notes about the quantitative genetics chapter before, and this is kind of the same deal, because I understand that in a single chapter about population genetics in a general genetics textbook, they're going to have a big range of material. Some of it's really mathematical, some of it's very difficult, some of it's very conceptual. There's all sorts of stuff in here. So again, please use the lecture as a guideline as to the topics that I have picked. I can't pick everything. And use the lecture as a rough guideline to the types of problems that I think are important. Okay, so we've gone through some problems. Some of those problems are easy, some not so easy. And on the test, I tried to make problems somewhat like that, but sometimes I try to add something so that you have some insight, right? But the lecture notes provide some idea about that. The book has lots of other concepts or equations, particularly terms that I'm not going to go over, so be careful about that, particularly for these selection things. The selection thing, we're going to handle it pretty much in this plug and chug set of columns, okay? I know in the book they have all these other tables about selection coefficients and so forth, but I'm not going to get into that. So, try to limit yourself a little bit on those things. And I've got a whole bunch of sections that I didn't go over. You're free to read this, but I didn't cover it. And those include forces that change gene frequencies in population. In that section, we don't cover mutation, drift, or selection in as nearly as much math detail as the book does, so be aware of that. We're not covering genetic variation in natural populations, summary of effects of evolutionary forces, role of genetics in conservation biology. All these things are interesting, they're important, but there are big sections in the book with lots of material, and I pretty much go through the lecture notes, even for those questions that I say, which of the following statements about blah, blah, blah is correct or false or something like that. Pretty much I skim through the lecture notes to kind of come up with those things. I'm not going to go find something obscure that we didn't cover in the textbook. So what should you be able to do? Things that we covered. Genotype allele frequencies, Hardy-Weinberg type problems. So genotype allele frequencies, we had those 3 approaches, right? You count them up, or you calculate it from genotype frequencies. Hardy-Weinberg problems. These are the classic problem that population genetics is known for. This is why it's important, right? You've given a certain number of individuals that are affected. What's the number of carriers? Those types of problems. Migration, combine some populations. That's a common problem. And these fitness selection problems to the extent that I've described in the lecture. Okay. So let's move on, okay? I have a few more topics here that I think are kind of interesting. It's amazing how much you can understand and how much in the world applies to genetics. And so here's some examples of things that I think are kind of interesting, not necessarily population genetics, but nonetheless, ideas related to things we've been talking about. And after we finish that, we will launch into bacterial genetics. So the first topic, unrelated topic here, is inbred organisms. So we've talked about cloning, right? Somatic cell nuclear transfer, where basically you create an organism that is identical to some parent from which you took a nucleus from. Inbred organisms is essentially a kind of cloning as well. It's done in a completely different way, but these animals are clones of one another, right? It's Star Warrior, if you will, right? So you're basically breeding animals together that are genetically identical. They're homozygous at every allele, and when you cross them, you get more of exactly the same thing. That's an inbred organism. The question is, why do we bother? What use is that? And I have a few ideas here. One is that if different labs use the same exact organism, the same genetics, then you hope that results in one lab will apply and be seen when it's duplicated in another lab. So that can be an important reason. The results tend to be more tight or more consistent when you use an inbred organism because the range of responses is generally a little bit smaller than if you have a wild type outbred line of mice, let's say. So you might look at the ability to metabolize some drug that's in a drug trial, and you would like to, get some idea about mechanisms and, how it's being cleared and all that type of stuff, and you would like your data to be as clear as possible. Inbred organisms are useful for this. Outbred organisms tend to show widely ranging responses to these types of things, and that would be something you'd have to worry about later on, but initially using inbred organisms is useful. So the results you see are less likely to be due to differences between individuals. You expect some consistency with these types of things. Outbred individuals have their own value. That means they're genetically very diverse, right? Outbred organisms are kind of what we've been talking about in population genetics, where you have a set of genes that all come in various allelic forms, and each allelic form has a certain frequency in the population. In outbred organisms, you've got a lot of choices at all these genes in terms of the particular allele that you might pick. And so the organisms that you can produce from, let's say, an outbred population of alleles is pretty wide-ranging. If you talk about inbred organism, you don't have any choice at any given gene that you pick out. You put your hand into the vat and pull it out, there it is. That's the allele that you get, and that's the only thing you can get, and you'll get that same thing over and over and over again. That's an inbred organism. So what does inbreeding do? Inbreeding, if it's done deliberately to create an inbred line for scientific research, for instance, causes original heterozygosity in founder animals, that is, diverse alleles at various frequencies in the population, to be reduced to homozygosity. So at every allele, every gene, you basically have the same sequence, right? So across all the genes in an inbred mouse, they have the same set of alleles as the next mouse, as the next mouse, as the next mouse, and so forth. A byproduct of trying to inbreed outbred animals to a inbred line is once in a while you start to see an increase of alleles that cause defects. So it doesn't always work, this inbreeding process. Sometimes you start to increase the concentration of recessive alleles that you hadn't seen before, and this causes the line to kind of die out. But it still can work, and that's why we have today a variety of inbred lines of organisms and mice and so forth. And I've got I think a list of a few of them here, and this is really pretty interesting. These are lines of inbred mice that people use in laboratory experiments across the country and around the world. This is BLAC6, BALB-C, DBA2. Each of these is an inbred line of mouse. They were selected for by parent matings and then sibling matings and then more sibling matings, generation after generation, to try to eliminate all heterozygosity. And if you look at the characteristics of these things, they're very clearly kind of individualistic, right? They have their own set of characteristics, and every animal of that type will respond or act in the same way. So let's see, BLAC6, black fur, aggressive mice, low testosterone levels. I mean, this isn't on the test, it just gives you an idea about what you get as a result of these types of inbreeding experiments. High alcohol preference, high activity on the exercise wheel, learn quickly in response to shock. Ugh. Would that work in genetics? Probably not. High locomotor activity, so forth. BALB-C, white fur, albino, visual problems, breeds well, low exploratory activity, low wheel activity, poor shock avoidance learning, low alcohol preference, a different set of characteristics. That's consistent with every BALB-C versus every BLAC6 and so forth. DBA2, pale brown fur, low alcohol preference, high wheel activity, males compete poorly for females, high incidence of mammary cancer, susceptible to seizures induced by loud noise, so on and so forth, right? There's all these things that people have studied about these things. What these are basically almost individuals that have been replicated over and over and over again to make enormous kind of research populations of animals, all of which share these different types of characteristics. So an interesting consequence of fixing a certain collection of alleles in one strain of mice, starting from all these possible alleles that you might pick from the beginning. Yeah, question? Yeah, in a certain sense, it's similar. I mean, they are clones of one another. You don't typically think of inbred animals as being clones. You just think of them maybe as a line of mice or something like that. But in reality, they are a kind of clone in that they are genetically identical. Okay, so what we're going to do is make an inbred organism. And I'm going to use the example of a plant because plants can self-fertilize, and this makes the process a lot easier. The same type of thing will apply if you were making a mouse, but there's a complication, a small complication, but the complication in the mouse is that you have male and female, and you have to breed them together, so you start with possibility of 4 different alleles and the 2 diploid chromosomes in the mouse situation. So we're going to start with a plant, and let's suppose that we have a plant which is heterozygous at big A, small a, and if we self-cross it, if that's something that's allowable in our hypothetical example here, what we would get as a result of a cross of big A, small a, is big A, 2 heterozygotes, and small a, small a. If we wanted to fix one of those alleles, A or small a, as being just a big A type or just small a type, and if we could observe the output, the phenotype of the A gene, then we could just look at the offspring and pick it, right? Maybe we can see that the A gene controls some phenotype, and we can say, okay, we pick this, we know it must be, homozygous big A, or we know the plant must be homozygous small a, small a. For that one gene, we've basically kind of inbred it in this simple example, right? We started with 2 possible alleles, but we end up with a line which will be true breeding at that gene A forever and ever. That's the idea of what we want to do across every gene in the inbreeding procedure. So the plant is true breeding or homozygous or inbred at that locus. The problem is for most genes, whether it's 20, 25, whatever thousand genes there are, we can't visually select for all of those, right? Most genes kind of act at the level of the cell or subcellular metabolism, machineries and stuff like that, and you don't see the output. It's only a few things that you can visibly select for and make sure it's homozygous for one allele or the other. But all the other genes, you have no idea. Right? You can't select for those. So that's the problem. We've got an awful lot of genes that we want to make homozygous, but we have no way of determining whether they are homozygous or not homozygous. So here's a kind of trick to that idea. Let's suppose our plant is big B, small b at some B gene. Now maybe we can't see the output of the B gene, okay? So we don't know what it is. But the worst case is it would be big B, small b. There would be 2 possible alleles if it's a diploid organism, right? So we don't know what it is, but we suppose worst case it's not already fixed as big B, or small b, small b. Worst case is it's heterozygous. A self-cross would produce homozygous big B, 2 heterozygotes, and small b, small b. So we make the cross, we look at the offspring, we have no idea what they are. We can't tell the output of the B gene, but we just pick something, right? Just pick it randomly. So you might get lucky, right? There's 50% chance that of the offspring, you would pick heterozygotes if we assumed we started as a heterozygote. That would just mean you got unlucky, okay? It's not fixed yet. But there's 50% chance that you would get big B, or small b, right? That's a pretty good odds for that particular gene. You have half a chance of fixing that B gene into either homozygous this or homozygous that, right? Let's suppose you fail, right? If you are unlucky and it just so happens you randomly pick an offspring and it's still heterozygous big B, well, do it again. Cross the plant again, Pick again, close your eyes and pick. You have another half a chance, like flipping a coin, of being either big B, or small b, small b. So you can apply this process just kind of automatically, saying that the output of any given gene that comes in two alleles, which is true for all the genes in a diploid organism, the chance that you're going to fix that gene into homozygosity of one type or another is 50% with every generation. Okay? Do you know whether you're going to get big B, or small b? No, you have no idea. Right? You can't see the output of this gene. It's just a gene that does something for the plant. Right? So you can't see it at all. You just rely on the statistics here to hopefully get yourself into homozygosity. Okay, so point is you have a 50% chance, coin flip chance, of selecting either of those two homozygous states. And we apply this notion across all the genes. Maybe if the organism starts with 30,000 genes, and if we, so this is our worst case, 30,000 genes, every one of them is heterozygous, maybe, okay, let's just suppose, because we can't evaluate the output of all those 30,000. You do a self-cross, and everything that happened at the B gene happens at those other 29,999 genes as well. And so you just pick an offspring, and you would expect that about half of the loci would now be homozygous, and half of the loci would probably still be heterozygous. You can't see anything, all you do is close your eyes and pick an organism. Now you take that one with roughly 15,000 homozygous loci, those are fixed. You can't get anything else out of that ever again, and 15,000 heterozygous loci. So you do the exact same process again. For all those loci that are still heterozygous, the next round, about half of them will be fixed into homozygosity, right? That is 7,500. And you just keep going until you reach this kind of point of diminishing returns where there's almost nothing left to fix anymore. And then you have an inbred plant line, okay, just by self-crossing and randomly picking. So this is kind of the idea of how you would kind of make an inbred line. The basic point is, after enough generations, you have obtained homozygosity at all the genes. You don't know which allele it was of the two that you started with, but you know that it's only one of those two possible starting types. Yeah, question? Yeah, I mean, you can evaluate that mathematically, but I mean, you could, I mean, if you do 7,500, you could keep going, right? And it would be what, 3,000 and 1,500 and so forth. So I don't know the exact number, but you can evaluate at what point you've got nothing else to do than just reading that inbred line. What's that? So the point is here that for most of the genes, you don't see a phenotypic effect, right? Most of the genes are just contributing to the growth and development and existence of the plant, right? You don't see anything spectacular like red leaves or blue leaves or whatever, right? You're just taking advantage of the fact that in these kind of Mendelian processes, this is the output of a gene that comes in two allelic forms. So you just close your eyes and you pick. And so you automatically are forcing homozygosity by half every generation. Okay, so the note here at the bottom is kind of an important note, and that is, if you started with an outbred plant, or even with one plant, and you did this procedure, you wouldn't necessarily end up at the same point if you did it twice, or if you did it in parallel, okay? So sometimes, when you did this and closed your eyes and pick, sometimes at some gene you'd get, let's say, big B, but you do it another time in another line of plants that you're doing the same thing, you might get small b. So across all of the genes in the final resulting inbred organisms, some of them will be, you know, big A, small b, big C, small d. If you did it again, you might have small a, small b, small c. You're not picking anything in particular, you're just forcing homozygosity at all these things. So if you did it twice and you went down and you got to the end point, the end point doesn't mean this plant is the same as this plant. They're going to be different inbred lines. And that's kind of what you see here with these mice too, right? You started with a particular, you know, parents, outbred parents, which had a particular collection of alleles in those genomes, and you applied this type of process, and you get out different results. You don't get the same thing over and over and over again, because you don't get to pick which direction you're fixing the alleles. Okay, so good. Just an interesting type of process and way of thinking about collections of alleles across the whole genome. So second topic here is just something kind of interesting or quirky, and that is hybrid seed. You hear about hybrid seed, what does it mean, and so forth. And if you go to a nursery or one of these home improvement stores, you can buy these packages of seeds, and they show you these nice pictures. This one is a carrot. It's something or other, Terra hybrid. This one is it a hybrid? Oh, luscious hybrid. Yeah, right, luscious hybrid. Hybrid grain X, okay? So these things are showing you supposedly what you're gonna get if you plant these seeds. And then the question genetically is, what does it mean, the hybrid part, okay? What else do you see here? Organic. I don't know why they can apply the term organic. They're just seeds. It's not necessarily something you're gonna eat directly, I don't suppose, but anyway. And they say guaranteed to grow. I don't know about that. Like, what's the alternative? You know, you hope they grow, right? Guaranteed to grow. And they show you these great pictures. Okay, so how does it work? Basically, hybrid seed, what these companies do is they'll take a true breeding strain of some plant, some organism, which has certain qualities, certain properties, and a certain kind of set of alleles, it's homozygous, and you cross it with another true breeding strain, another kind of inbred strain that has certain qualities or properties. And it turns out sometimes when you cross organisms like this, one variant or strain versus another, you generate a hybrid composed of the genomes of both of the parents, and you get a lot of the qualities of both of the parents. And so there are some agricultural advantages to these hybrids. Their growth tends to be robust. Whatever they produce tends to be, larger, things like that. So there are some advantages in certain cases to making these hybrids. So they will cross strain A, which is true breeding, against strain B, which is true breeding, to generate F1 seeds, which are AB hybrids, and that's what they're selling in those packages, essentially, okay? And there's some advantage to that. They also, they charge you money for doing this, but they also charge you money for marketing, right? And to make these pictures and make it look nice and appealing and so forth. Okay, so here's a question about the hybrid seed. Can you kind of avoid the company next time around? Can you get the F1 seed, plant the seeds, grow up the plants and harvest some, and let other ones make seeds, and take the seeds and do it all over again without having to go back and purchase more of these hybrid seeds? And it's kind of a difficult question. So if you think about it, here's the question. If you grow the F1 hybrids, collect the seeds, which is the output of an F1 times F1 cross, at the end of the season in the replant, can you bypass the company next time around, or do you still have to buy seeds? So if you think about it, the F1 hybrid, the seed that you're getting from the company, you plant that, it has a genome which is part strain A and part strain B. So it's basically heterozygous for the alleles that made strain A have some characteristics, and the other part of it is the alleles from strain B that had some characteristic. You take the F1s, you grow them up, and they're going to flower and all this stuff, and you let them go to seed, which is basically an F1 times F1 cross. Do you get the same thing all over again? So if you think about it, the F1 is heterozygous for parental strain A alleles and parental strain B alleles. But during meiosis, what happens is you mix everything up. A and B alleles get all mixed up, right? We've seen this again and again, where grandma's delivery and grandpa's delivery in a parent is all shuffled, and then the offspring gets some combination. That's what's happening here. And the gamete that you get as a result of meiosis in the F1 that you've grown up is mated to another gamete, a pollen or whatever, from some other F1, and so you get a really complicated mix of the A and B genomes in the seeds of these F1, F1 crosses. So as a result, plants produced by these F1 hybrid crosses really display a huge range of qualities, a lot of which aren't that appealing or productive and so forth. So it tends not to work. You might get lucky, but the chance that you get lucky is just so exceedingly low that you would still kind of retain a collection of all the proper alleles. So the F2 are unlikely to retain a sufficient collection of the original A and B genomes that gave their hybrid parents their superior characteristics. So you let all these F1s go to seed, you collect it, you plant them next year, you don't know what to expect. So it's kind of an interesting idea having to do with the breeding of these things and things that you can buy in the store, have some agricultural value. Okay, are we good so far? Yeah. Inbred versus true breeding. So true breeding can apply to one or just a few genes, right? So you've seen enough of these problems where it starts out a true breeding parent for such and such gene, right? If you mean inbred, really you're talking about the whole collection. That's kind of the distinction. Okay, so we'll start bacterial genetics now, and this is the last section prior to the exam on Tuesday. I don't plan to get too far. We have to leave something for Thursday, but we'll start out with some background material and then start to talk about some mechanisms by which bacteria transfer genetic information from one cell to another cell. Okay, so bacteria, it's completely different, right? Everything that we've talked about so far, most of that stuff kind of doesn't apply in the same way in bacteria as it does in eukaryotic organisms. There's no nucleus, right? There's no nucleus. This is a common mistake people make when you talk about bacteria and eukaryotes. It's easy to say, well, in the nucleus of these E. coli cells, but it's not true, okay? So you want to kind of guard against simple stumbles like that. There's no nucleus in these, no subcellular organelles, no mitochondria, all that kind of stuff. So completely different kind of life form. The model that we use in laboratories is most of the time E. coli, Escherichia coli. And here's a depiction of a plate of bacteria cells that have been spread out on this on the agar surface. And in the agar is some media that allows the cells to grow up, sugar, salt, vitamins, and so on and so forth. And I don't know whether you have ever done one of these in a laboratory course, but it takes a little bit of practice, right? Because what happens is you take a toothpick or whatever and you dip it into some bacterial cells, and then you take your new plate, and what you're supposed to do is streak this way, right? And then you use a little bit and you streak this way, and then you use a little bit and you streak this way, and so forth. And you're supposed to go from just densely packed cells that almost form a lawn here to individual colonies. This is what you want to do when you streak out a plate like this. You want to be able to say, okay, I can now pick this one or this one or this one, some individual, right? That's what your goal is usually in these things. But it takes some practice. So what happens is usually when you start out, you come the next day and look at your agar plate and there's nothing there at all, right? That happens. Maybe you burn it, you sterilize the loop, and so you dip it in and they're all dyed, and so there's nothing there. Or you get a big blob of cells right here and there's nothing else on the rest of the plate, right? Or most often you streak a bunch of stuff here, you streak a bunch of stuff here, you streak a bunch of stuff here, and it all looks like that. You never get to that kind of dilution point where you've streaked it out to form individual colonies. So, have you done it? Who's done it? A few people in laboratory prep. Did it work? No. Yeah? Okay, good. All right. Okay, so other feature of E. coli, circular chromosome. I think it's about 5 million bases or base pairs long, and there's something like 4,000, 5,000 genes on it, roughly, 4,500. Circular chromosome, not linear like in nuclear DNA of eukaryotes, and very, very much larger than the mitochondrial circular genome that we've talked about so far. The human mitochondrial genome is 16,000, 18,000 base pairs, something like that. This is a lot bigger circle than that. So 5 million, and of course, human genome is 3 billion, right? So we're 1,000 times bigger in our genomes compared to these genomes, just as for relative comparison. It's easy to grow these things in liquid media or agar plates. You can inoculate LB broth at night and come in the morning, and you'll have a cloudy tube in which your bacteria has grown up. And so it goes, 1, 2, 4, 8, 16, some sort of exponential growth pattern. And if you grow E. coli in the best possible conditions, it's roughly 20 minutes per generation. This is one advantage of studying, let's say, genetics of bacteria, is that you have the ability to screen for so many different progeny in a reasonable period of time. Mice take weeks, right? But E. coli, overnight, you get so many generations. And so you can screen for lots of different mutations around the genome and assay them by growth conditions and so forth. So here's a growth curve of a typical bacterial culture. The idea would be at time 0 here, you inoculate some liquid LB media with some cells, and the cells initially are kind of asleep, as it were, and it takes them a little while to wake up. That's the lag phase. But once they get going and realize they've got everything that they need, they really explode in this log phase or exponential growth. You see the axis here is log 1, 5, 10. So they just go crazy. And then eventually they slow down because the population becomes so crowded and nutrients become limiting and so forth. So this is almost a kind of going back to sleep. And then you can take these and you can do this all over again. And like I said, the advantage for genetics is just the number of offspring that you can screen in a short period of time is just phenomenal in comparison to the typical organisms we looked at so far. Okay, so what we'll talk about in terms of bacterial genetics is transfer of genetic information through several processes like conjugation, transformation, transduction, and we'll look very briefly at bacteriophage genetics. We're not going to cover fine structure analysis. Fine structure analysis is in the book, either in this chapter or maybe in a separate chapter. But in fine structure analysis, what this guy, Seymour Benzer, did was he looked at recombination points that were very, very, very close together. So recombination that occurs even within a gene and recombination between nucleotides that are very, very close to one another. So he used recombination as a way to kind of map the structure of genes. And it's neat stuff, but nobody really kind of does that anymore. We do sequencing, we kind of know the structure of genes and so forth. So I'm going to limit our discussion to these topics here and not go into fine structure analysis. Okay, so the first one, probably the one we'll cover today, is conjugation. So here's the experiment that kind of led to the discovery of this system. The idea is you have a strain of bacteria growing here and a strain of bacteria growing here, and each strain has its own genetics, its own genotype. This strain has a genotype that's indicated here, and in E. coli we often use some 3-letter name that describes something about what the gene does. Right? So in this case, this strain is 3+, Lu+, 5+. What that means is that the gene, or the E. coli has wild-type functional copies of genes involved in the metabolism or biosynthesis of threonine, leucine, and thymine. Okay? But it's minus or no plus for met and bio. These are other genes involved, let's say, in methionine biosynthesis, and they're defective. So not having a plus means these are defective. In this particular case, that would mean this strain in minimal media with just sugar, salts, and essential vitamins would be able to make threonine, would be able to make leucine for its own purposes like translation and so forth, but it would not be able to make methionine because there's some defect in that pathway. We saw the methionine pathway before where we had those kind of growth charts and we mapped a series of metabolic steps based on the growth. So that would be, this would be some step in that kind of a pathway, failure to make methionine. The strain B is kind of the opposite of strain A. It's plus for met and bio, but it's minus for the other three. So this one should not be able to grow on minimal media. If you took some out, plated it on agar plates that have minimal media, it won't grow because it can't make these two compounds or these two components. And this one, if you plate up minimal media, it won't grow either because it's defective for 3-Lu-Thi. Okay? But the kind of mystery is you put them together into one tube, and after a while you take out some cells and you grow those cells on minimal media, and lo and behold, somehow they are able to grow on minimal media. So apparently, at least one explanation would be there had been some sort of transfer of genetic information from this strain to this strain, or this strain to this strain, in such a way to create a new strain of E. coli that were now wild type for MET, bio, 3-Lu, and Thi. So that's one possible interpretation for what happened in this experiment. Another possible interpretation would be, well, wait a minute, you have some cells that can make some things, and some cells here that can make some things. Maybe this is just a complementation kind of an assay, where you plate everybody out here, and whatever each strain is able to make is kind of diffusing throughout the plate. So even though these guys can't make MET, maybe they're getting it on this plate because these guys can make MET. That's at least a reasonable explanation. Okay? Now we know what the mechanism is, and it does involve genetic transfer of genes in one direction or the other direction. So we know that in fact these colonies are a new unique genotype where they're plus at all of these markers. So how does it happen? Here we have another variant of this same experiment. In this case, what we do is we put strain A on this side, we put strain B on this side, but we separate them by this filter. The filter is some mesh that has really, really, really fine pores. Maybe the pores can allow amino acids and media, liquid to transfer back and forth, but you can't have cells moving back and forth. So this separates strain A from strain B such that there's no contact between them. And when you do this and take out this and take out this and say, can it now grow in minimal media? It can't. So strain A in this experiment stays like this, and strain B stays like this. You never get colonies that are of a new genotype. And the conclusion from this experiment is that contact between the two types of cells was required in order to transfer these genetic markers from one to another. Okay, so we know how this works. The mechanism involves a separate piece of DNA called an F factor, or fertility factor. This is a piece of DNA that almost confers a mating type-like aspect to the E. coli cells. So we can have this F factor. The F factor will form a bridge or a pili from one cell to another, and then across that we can transfer DNA to convert the genome of a recipient cell to something new that it wasn't when the experiment was started. Okay, so we can have several different types of bacterial cells that are going to be different with respect to the disposition of the F factor. So one type of cell would be those that do not have F factor DNA at all. Those are F minus, so they are of, let's say, a particular mating type almost. We can have cells that have an extra chromosomal copy of the F factor. So the F factor could be a circular piece of DNA that can replicate by itself independent of the host circular chromosome. So you'd have the host chromosome and you'd have an F factor that's doing its own thing outside of the host chromosome. And cells of that type would be called F+. And we can have cells where the F factor has integrated into the host cell genome and is kind of propagating or replicating along with that genome during DNA replication, just as if it were another gene, okay? So these cells where the F factor DNA is integrated into the genome are called HFR for high frequency of recombination. So these are the three kind of mating types, so to speak, that we can have as a result of this F factor. The point here is if the extra chromosomal F factor in an F plus cell moves into the E. coli host DNA, it will then go from F plus to HFR strain. Okay, so what types of matings are allowed and which ones are disallowed? F minus against F minus doesn't happen. There's no F factor, obviously, and you can't form the bridge between cells, so nothing happens. F plus times F minus is allowed. We can think of these as two mating types, and the output is both cells usually turning into F plus. One originally was, and now the other is converted to F plus, so that's the output of F plus times F minus. HFR strain times F minus, this mating is allowed. The F factor is integrated into the host DNA, but it still encodes all of the proteins necessary to make the bridge and so forth. So we can have an HFR times F minus mating. And this is what's important in the explaining conjugation, is that when the F factor moves over from an HFR strain to an F minus strain, bacterial genes come along with it and convert the recipient from its genotype to a new genotype. Usually in this case, the initial cell will remain as HFR and the initial recipient cell will remain as F minus. You don't transfer all of the F factor in these types of matings. F plus times F plus doesn't occur. Being F plus kind of immunizes yourself against this genetic transfer. Same way HFR times F plus doesn't occur, and I added one, HFR times HFR shouldn't occur. So being F plus or HFR already kind of immunizes yourself against transfer from some other donor, but if you're F minus, you can receive genes, either F factor genes or bacterial genes by these matings of F plus F minus or HFR F minus. This summarizes what can happen. So in picture form from the book, we have this notion of what's occurring during an F plus times F minus mating and an HFR times F minus mating. So I think F plus times F minus is in this column, and HFR times F minus is in this column. Okay, so for this one, the idea is you have the bacterial DNA, circular DNA, and you have the F factor DNA. The F factor DNA is extra chromosomal, right? It's outside of the genome. And this isn't to scale by any means, right? The host chromosome ought to be enormous relative to the size of the F factor, but it's just a drawing. And what happens is we form the bridge between the F plus and the F minus. The F factor DNA gets nicked at some point. This is what it's supposed to do. And it kind of spools its way across into the recipient cell. So what happens in this case is that we spool across usually a complete copy of the F factor DNA, and that's why at the end of this type of conjugation experiment, the donor is still F plus, but the recipient now has been converted from F minus to F plus. Okay, so there we have kind of a change in mating types. In the case of the HFR times F minus, that's more interesting, and that's the kind of problem that we work here. Here the idea is you have an F factor, which is outside the chromosome, but it integrates into the host DNA at some point, and the point can be random. It can be pretty much anywhere around the circle of E. coli genomic DNA. So we have a recombination event that occurs randomly somewhere, so that now the F factor is part of the host DNA, and it will replicate just as though it's another gene or a few genes in the host chromosome. But what happens is the HFR strain, as it's called here, has the capability to mate with an F- strain, which doesn't have the F factor components. So we get the same type of thing happening here as we saw over here. We have the bridge form, we have a nick occur in the F factor DNA just like it's supposed to, and now we start to spool across a strand of DNA of which the first part is going to be F factor, but later, because there's a directionality to this, you start to bring in nearby parts of the bacterial chromosome. So you move over stuff, part of the F factor, and then afterwards, the part of the donor chromosome itself, okay? And it's moving over this donor part that allows conversion of whatever this genotype is to replace whatever the original host or recipient genotype was. Right? So that's the way that you would have conversion of one of these to the other one, or back again, depending on which one was HFR, to form a new genotype here. Okay, so let me see if I've said everything. If F factor is integrated into the genome, it can bring cellular genes along with it when it's transferred. That's what's happening here. Only a single strand is transferred. Then what happens is some sort of recombination event between the transferred strand and the recipient genome, or maybe we convert it to double-strand DNA, then we have some recombination event. We're kind of waving our hands and not worrying about the mechanism here, but just to say that DNA kind of likes DNA and things interact to convert one genotype to another. Genes are transferred starting at where the F factor is integrated, and it depends on the direction. So the idea here is that it starts here and it pulls in genes from this side in this example, not from this side. And I've got a few more notes. Oh, the time it takes for a gene to be transferred, that is, the time it takes for a recipient marker to be converted to a donor marker in one of these conjugation experiments is timed in minutes. And so you can use this timing in minutes as a way to evaluate gene distances along the E. coli chromosome. And people would do this, and genes would be separated by certain numbers of minutes. And by this criteria, the entire chromosome was about 90 to 100 minutes long. If you could have a perfect conjugation experiment where the bridge was intact for that long of a period of time, which may not be likely, but if you could, the idea would be the F chromosome would be nicked and it would move over, and as long as that was stable, you would just keep moving stuff over and over and over again until you moved kind of one strand of that whole chromosome back over. Okay. So, what do we do with this? Basically, what we can do with this is map the positions of genes along the E. coli chromosome, and we can get some idea about how different strains of E. coli can share genes amongst one another to convert a genotype to another genotype and so forth. So in this slide, we have the same basic idea happening in terms of conjugation and gene transfer that we had before. We've just made it a little more complicated because we have added the location of certain markers that we're going to assay for when the DNA moves across this bridge. So we have a recipient cell, F-, which has certain markers or genotypes on the chromosome. And we have a donor cell, which is HFR, and we can see that we're going to have transfer of the little bit of the F factor followed by whatever donor genes are behind it. And we're going to have transfer of those sequences over into the recipient cell. Okay? At very short periods of time, you're only going to transfer sequences that are very near the location of F factor integration. But if you wait longer and longer, if that bridge is still there, you would have sequences being transferred that were farther and farther away from the site of F factor integration. Right? So we can time how long does it take in minutes for a particular marker to move over and then convert the recipient to a new genotype. So in this particular case, I can hardly read it, but we have we start a conjugation experiment. We allow transfer of genes for a short period of time, and we see The 3+ and LU+ markers convert the recipient from 3- to LU- to 3+ LU+, and that happens very early. That tells you the F factor is located very near the 3 and LU genes. If we wait longer and longer periods of time, we start to see other genes being passed through. Azide resistance, TUN-R, I'm not sure what that is. It might be another antibiotic resistance. Is that LAC+ maybe, GAL+, and so forth. So with time, we see farther and farther markers transferred. So this is basically the concept of using this procedure to map the locations of genes. Yeah, question? Not as far as I know. I mean, usually for these types of things, it's a deliberate process. It's machines just kind of running, and they do a certain thing. It's not impossible, but there's no deliberate mechanism. There's no machinery in the F- necessarily to take advantage of it and send it the other direction. If it did, it would be kind of an accident, right? But there's probably some directionality to how the equipment is set up, some conveyor belt that moves in a direction, something like that. So you don't necessarily have to break it deliberately yourself. If you have a tube of cells with 2 types and you set it up, there's a lot of motion. You don't know there's a lot of motion because all you see is a tube of liquid. But if you're a cell, things break apart. Yeah. In fact, it's very hard almost to not break. That's right. So you could deliberately, what you're doing is you're setting it up at time 0 and you're taking aliquots, right? And that breaks it. You're taking aliquots and spreading them out. And so you've deliberately said, okay, at this time, what do I see in terms of growth characteristics? Then you wait longer and you take another aliquot and then you do that again. Okay? So if you were to then analyze this data and say, what do we see, what do we get out of this? We would see in terms of a map where we put markers against minutes it took for conjugation to occur. We would say, well, at some point, that's where the F factor was, and it took us, 7 or 8 minutes to get these markers, 3 and LU through. It took us 9 minutes to get azide, 10 minutes to get this TUN-R resistance, 16 for LAC, 25 for GAL. So that positions the order of these genes along the E. coli chromosome, and that's one of the values of this in early bacterial genetics studies. And a table or a graph of this idea is shown here as well. You have the time in minutes where the conjugation occurred, and you have the appearance of recipient cells of a particular new genotype occurring at various times in minutes. So at 8 or 9, we have the first few. At 10 or so, we have this TUN gene, and blue is LAC, and GAL is at about 25. So this is just the same thing as we have here, but we have some other data here, and that is the efficiency of transfer. And the idea is, when you set the experiment up, you have, let's say at time 0, 100% maximum conjugation that you're going to have, right? So as many cells as possible have undergone conjugation and are transferring markers. So the efficiency of the early marker transfer is very high. You see a high proportion of the recipients then being converted from one genotype to another because the genes were so close to where the F factor was integrated. But if you wait longer and longer times, a lot of these bridges have broken apart, and there's fewer still kind of transferring DNA through at longer periods of time, like 25 minutes. And so you see that the efficiency of the GAL+ marker transfer in the recipients is much lower than those initial markers. So we see a couple of things here. One is the time that it takes for a particular marker to transfer, and the other is the efficiency with which that marker transfers based on how far away it is from the F factor integration site. Okay, good. So the graph shows genes near integration site are transferred first with high efficiency. Genes farther away from the integration site are transferred later and have lower efficiency due to interruptions of batings. And in the experiment, this experiment here, we're transferring azide resistance, ton resistance, black plus, gal plus, same as the previous experiment, but I guess the first two have been omitted. If this is what's transferred, you might suppose that the F factor in this experiment is different from the F factor location in this experiment. In this one, we have seen pre and Lu marker transfer. In this one, we haven't, okay? Assuming that we're looking for those markers. Another point is at the end of the experiment, you only want to count the recipient cells. You want to kind of have some way of getting rid of the donor cells, right? Because the donor cells, in many of these cases, are plus for something. So if you have donor cells and you're growing them on your media to see what happened, they will grow. I mean, you'll be able to see that they're plus. What you really want to assay for is the conversion of the minus marker to the plus marker. And you want to be sure that you get rid of all the donor cells. So there has to be some trick here where maybe the recipient cells are resistant to some unique antibiotic. You could apply that selection, and then of the colonies that you see grow up under various growth conditions, you know that you're only looking at recipient cells, and you have cleared the system of the cells that were the donor cells. Okay, so what do you do with this? Typical problems will give you a series of HFR strains where the F factor is integrated at various locations around the E. coli chromosome, and you'll be given some data about, marker transfer order or the timing of transfer order or something like that from these different HFR strains. And you're basically supposed to overlap everything and create a map, right? So, you get ABC, BC, D, can I do the alphabet, C, D, E, you overlap everything and you get A, B, C, D, E. That's the idea. Only they kind of, they'll shuffle them up so it's not A, B, C, D, E, F, G. That's kind of what you're supposed to do with a set of HFR strain marker transfer experiments. And in those, you have to realize that the position, so here we have a kind of pretend genome, A, B, C, D, E, F, G, H, I, J, all right? And in this pretend genome, or in this gene order, this F factor has inserted or integrated itself to form an HFR strain between the E and the F genes, right? But in another HFR strain, the position will be different. In this one, the F factor has integrated itself between the C and D genes, right? So that will be one, usually a unique aspect of each of the HFR strains that are used in these mating experiments. Other point, F factor can be inserted in different orientations. So here we have a set of genes or regions, A, B, C, D, E, F, G, and the same here. Maybe these are two different HFR strains, that's fine. And we notice the F factor is located between D and E in this strain and between D and E in this strain. But they don't have to be the same because the orientation of the F factor in one of them can point one direction and then the other one can point the other direction. So maybe in this HFR strain, if we were to examine marker conversion in the recipients, it would be F going through, or part of F, and then DCBA, so the orientation would kind of pull things this way. And in this one, maybe the F factor is oriented differently, so it pulls E, F, and G with it, right? So the orientation of the F factor is important in determining whether it's going to pull genes from one side or the other side. And the other thing I think I forgot to mention is, why don't you convert HFR to F plus when you pull these genes across? And the reason is, you nick the F factor somewhere inside it, somewhere internally. So while you get a little bit of the front part of the F factor moving over, you don't get the back end, which is all the way around the E. coli chromosome. And that's why it's unlikely that you would convert HFR to another HFR to F plus or something like that. Probably you're going to stall conjugation at some point and you'll only transfer partway around the E. coli chromosome and don't get the other end of the F factor. Okay, I think I already mentioned this. So typical problems will ask you to generate a map from a set of HFR strains, A, B, C, D, E, G, H, I, J, G, F, E, D, C, B. It's not that hard. Well, it can be. This one's not that hard. You just kind of line everything up to regenerate. In this case, it's the alphabet. Okay. So things that you might think about or worry about. would be, can you make a circle, right? If you're given a set of HFR strains and marker transfers and so forth, can you link all the genes all the way around in a circle, or is it just a line and the ends are kind of, you're not sure what's going on, right? So you could make pretend problems where, well, the chromosome was linear, right, because you can't put them together. Or you don't know whether the chromosome is circular because you can't put them together. Like in this case, the issue is, well, does A connect with J? We don't know. We don't have information about that, right? It could be, it's like this, some organism from Mars or something, it's like this. Or maybe it really does connect, but we haven't assayed for everything else around here and so forth. Or maybe you have two circles. Here's one, and maybe we could make a circle out of it. And maybe there's some other HFR marker transfer experiments that form something else, you know. So maybe you have two units that you can't connect. So you could make little puzzles like this. Nothing really is different from the basic idea, though, that is you get a series or an order of transfer and you try to put them together and then interpret what that means. Okay, so that's it for today. We will finish up on transformation, transduction, bacteriophage genetics on Thursday.